Leggity low, my beggity boys and giggity girls. Alright, today we're gonna cover enzymes 4 hours until the exams. How we feeling? How we feeling, people? Alright, so, let's get into this quickly. First of all, what is an enzyme? We'll take lipase as an example here. Lipase is a catalyst for digestion. For the digestion of fat, right? What is a catalyst? A catalyst is basically a chemical or material which helps one material react with a different material. For example, my friend slowly pulling away from me for multiple months on end is a catalyst for me wanting to kill myself, or me scratching my ass too hard when wiping is a catalyst for my ass bleeding, right? So here are the 3 main types of enzymes that will be covered in the exam. We got protease, carbohydrates, and lipase, amylase, lipase, carbohydrates, and protease. Right? Lipase breaks down fats into glycerol and fatty acids. I remember this because my mate when we were in year 7, 8 was talking about using fat and then digestion to make nitroglycerin or something. Teachers overheard him. Then he said I was the one doing it instead. I mean you gotta do what you gotta do, but still kind of feel, But so carbohydrates breaks down big sugars into small sugars, right? And protease helps digest proteins into amino acids, right? First, you might be wondering how do these enzymes work, right? So this is lipase and this is fat. You see, enzyme substrate, right? You can see that there's this area where they match perfectly like a jigsaw puzzle, right? This is called the active zone, and it's where one enzyme and one substrate perfectly lock into each other. So this means that only one enzyme can lock onto one specific type of substrate. You see? This is amylase, a type of carbohydrate, and this is the same fat molecule. This is a different type of fat molecule, I think. I don't know. Alright? You can see that they're not the same. So thus amylase or carbohydrates cannot digest fats. And there's an optimal temperature and an optimal pH, optimal meaning best, for all of these, right? But you've probably heard that in class. There's a temperature and a pH, i.e. acidity, alkalinity, in which these enzymes work best. For example, in protease, it's in the conditions of the stomach where it works best. You might be wondering what happens when it leaves the conditions of the stomach, or strays too far away from the conditions of the stomach. That's called denaturing, which is actually a permanent process. See, this is a protein. This is pepsin, which is a type of protease. All these drawings are very oversimplified, by the way. And this is denatured pepsin, and this is the same protein molecule. As you can see, it won't fit into it anymore. You may be wondering where you can find all of these. Lipase is made in the pancreas, and it's injected into the small intestine immediately after the stomach, along with bile. Bile is actually alkaline, and it neutralizes the stomach's acid. So lipase works in more alkaline or neutral conditions, such as like pH 8, 7 to 9, Carbohydrates is specifically amylase can be found in your saliva, and so conditions of your mouth, i.e. a more neutral or slightly acidic pH, and the temperature, and like the body temperature, normal body temperature is probably the most optimum temperature and pH for carbohydrates. And as I said, protease is especially pepsin can be found in the stomach. So it's best able to work within conditions of the stomach, i.e. high acidity and a temperature of about 37 to 40 degrees Celsius. In conclusion, enzymes help with the digestion of certain molecules, and they do this by locking onto these molecules called substrates, breaking them down into simpler molecules, such as complex sugars into small sugars. They can only digest one type of substrate, and they have specific conditions that they need to be in, otherwise they'll denature, which is a permanent process and cannot be undone. Alright, good luck, God bless you all. Good luck in the exam, people.